Hello, everyone, and welcome to today's live broadcast, Strategies for High-Titer Protein Expression using the ExpiCHO and Expi293 Transient Expression Systems, presented by Jonathan Zmuda, Director, Cell Biology, Life Sciences Solutions Group, Thermo Fisher Scientific. I am Alexis Krauss of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots and sponsored by Thermo Fisher Scientific. For more information about our sponsor, please visit www.thermofisher.com. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. We'll answer as many of your questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, please use the Ask a Question box to let us know that you're having a problem. Please join me in welcoming Dr. Zmuda. I will now turn the presentation over to him.
Thank you, Alexis, and good morning, good afternoon, and good evening to all the attendees. Thank you for joining us again for our ExpressionWorld event. And as Alexis mentioned, today I would like to talk about strategies for high-titer protein expression using our mammalian ExpiCHO and Expi293 expression systems. The Expi293 expression system was first launched about 6 years ago at this time, and ExpiCHO almost 3 years ago. And during that time, we've been able to put together a broad set of data regarding the best strategies for utilizing the CHO versus the 293 expression systems. And in this presentation today, I will go over some of those strategies as well as the differences and similarities between the protocols for the different expression systems. So I'm sure that it comes as no surprise to anybody on the call today that biologics today comprise 10 of the top 15 selling drugs, and you see a list of them below. So certainly within biological development, protein expression has exploded over the last couple of decades, and certainly even for those small molecule compounds that are not themselves proteins, the requirement for protein has continued to increase during the drug development process, even for the small molecule reagents. So when we look at the usage of mammalian cell lines in the biologics development process, many years ago, the HEC293 cells were the workhorse of all protein expression, partly because they were easier to transfect and you could get reasonably decent protein titers in the 293 cells, whereas the older generation CHO-based systems had very low protein titers, often in the 10 to 20 milligram per liter range for monoclonal antibodies. With the advent of Expi293, we were able to increase the protein titers from 293 cells in the range of 3 to 5-fold, or in many instances even greater than that, which allowed for the utility of the Expi293 cells to be greatly enhanced and provide more protein for researchers' needs. About 3 years ago, as mentioned, we launched the ExpiCHO expression system. And the ExpiCHO transient expression system allowed for even higher titer protein expression than Expi293. And as you see towards the bottom of the screen, to date, Expi293 is often being used in the very early discovery, target ID, candidate generation stage in biologics development, whereas ExpiCHO is tending to span the entire development stage, often from discovery all the way through material for animal trials. And in the bottom right, you see that very recently we have launched an ExpiCHO stable expression medium that allows for large-scale production of stable ExpiCHO clones for clinical manufacturing purposes. Later this year, we will also be releasing the GMP cell banks, as well as we have late last year already released the GMP Expi293 cell banks available to customers. So when we look at the three different expression systems that we have, and on the far right, I will note one of our newest expression systems, the Expi SF expression system based on SF9 cells, that was released just a couple of weeks ago, we now have 3 high-titer expression systems between Expi293, ExpiCHO, and ExpiSF that cover a broad range of the typical host cell lines used for protein expression, with the paradigm being that for any given protein that you need to express, we would hope that one of these high-titer expression systems would be available to provide that protein for you. And again, I will go into a bit about the paradigm in terms of which proteins tend to be expressed in which systems. So if we look at an overview of the Expi293 system, so this of course is a human HEK293 based expression system with typical antibody yields up to 1 gram per liter. And the focus historically has been on antibody screening and significantly also reagent and immunogen generation, proteins for structural biology, membrane and intracellular proteins, and AAV. And again, what's new with the Expi293 system as of today is that last year we did launch the GMP cells for Expi293 available for licensing currently. When ExpiCHO came on the market, ExpiCHO was able to dramatically improve the protein titers seen in CHO-based systems with antibody yields up to 3 grams per liter or even higher in some instances. Typically, ExpiCHO has been focused on monoclonal antibodies, Fc fusion proteins, FABs, bispecifics, and some vaccine work as well. And generally, ExpiCHO seems to do a very excellent job at expressing anything that is antibody or antibody-like, including bispecifics and multispecific moieties that are very complex. It seems as though the CHO cells have a bit more diversity in their ability to express some of these atypical molecules. Similarly, also, certain species of antibodies, including most specifically mouse and rat, do not express very well in the 293 cells, whereas the CHO cells do an excellent job and can, again, generate titers in the gram per liter range for the mouse, rat, and rabbit antibodies that we'll show some data for a little bit later in the presentation. So again, what's new here, as I mentioned, is that later this year we will have the ExpiCHO GMP bank cells, and we've been able to demonstrate that compared to the parental CHO-S cell line, that we can typically see 2 to 4-fold greater titers in the stable ExpiCHO clones, often reaching into the 3 to 4 gram per liter range, and pairing that with our large-scale stable ExpiCHO media that's available also in an AGT format for the time when you are ready to scale up into larger scale. The stable production media will not support transfection of the cells, so it is in fact made for the expression of the stable clones, but for all cell line generation work, the ExpiCHO expression media would still be utilized as it is very compatible with transient transfection. And I will talk just very briefly about our new ExpiSF system. So Maya Yovcheva from my team will be giving the next talk and going into great detail on the ExpiSF expression system. But we're very excited about this system because for the first time in the insect world, we have developed a chemically defined and completely yeastolate-free media that allows for high density SF9 cell growth and very excellent lot-to-lot consistency now that yeastolate has finally been removed from the insect media. So if we think about this, it is somewhat akin to serum being removed from mammalian media many years or a decade ago. We now have a completely defined yeastolate-free media for the ExpiSF system. When we compare this to our standard SF9 system, we typically see protein yields 3-fold or higher than the standard SF9 system. And what we've seen to date is that there's a significant focus on both intracellular proteins as well as proteins that may be toxic to mammalian cells, and certainly a very large play around vaccines, VLPs, and AAV being expressed in the insect system. So again, following my talk today, there will be a full session on the ExpiSF system, so please join if you have interest also in introducing this third Expi system into your laboratory. So what makes the Expi systems unique? So there are many quote-unquote systems out there for 293 or CHO cells, most of which comprise at most a transfection reagent, possibly a media as well. However, to truly take advantage of high-density transfection and elongated protein expression runs, one must use more than simply cells, media, and transfection reagents. Because in those instances, if we were to transfect at a very high density without controlling those cells post-transfection, the cells will simply continue to grow, they will reach their peak density, and they will die off, and you lose all of the associated benefits with that additional biomass of transfecting at 3, 5, or 6 million cells per mil. So really what the Expi systems comprise is a fully developed system of cells, media, transfection reagents, our proprietary expression enhancers and feeds, as well as the DNA vectors to go along with them to optimize protein expression. And it's only when all of these components are brought together in this systems type approach that we can truly get order of magnitude increases in protein expression compared to any other systems on the market. So what I'd like to do now is talk a little bit about both systems side by side. So for the last few years, we've spent a lot of time talking about ExpiCHO, while Expi293 has continued to be a mainstay in many laboratories. And what I'd like to do in this presentation is focus on both of the Expi systems, compare and contrast, and give some tips along the way for each step in the protocol. So if we look at the two different cell lines, on the left we've got the Expi293F cells, and of course these are human cells derived from the freestyle 293F cells. They are adapted for high density culture in the Expi293 media, so these cells will typically attain about 15 million cells per mil peak density. Their doubling time is about 24 to 25 hours, and the cell diameter is fairly large at about 18 micron, and these cells, similar to the ExpiCHO cells, will become larger, visibly larger, during the expression runs when they're producing high levels of protein. The transfection efficiency in the Expi293 cells is very high, so 80 to 85% is probably even a bit of an understatement. Oftentimes it can be 90% plus. But I do want to point out here that transfection efficiency is not nearly the most important part of these systems. So while it is important and it is one of the pillars, simply getting the highest transfection efficiency does not necessarily correlate to the highest protein production. Once again, because it is the downstream aspects of the systems that allow the cells to continue to remain highly viable and generate significant protein over many days. The ExpiCHO cells over the years have been shown to have an exquisitely stable growth and expression profile, at least to 30 passages, and many times laboratories carry them out even further than 30 passages, and we get very nice high-quality biologically active protein. So on the bottom you can see in the middle pane what the cultures look like at 5 million cells per mil, and then to the right pane 12 million cells per mil. Should see essentially no clumping of the cells, even at very high densities. So that is always something to keep an eye out for, that if cells are clumping, oftentimes it can be indicative of culture conditions that are suboptimal and perhaps also temperature conditions within the flasks, or even perhaps lack of recovery from a particular cell bank. So contrasting that to the ExpiCHO cell lines, so these are a subclone derived from our GMP CHO-S cell banks, chosen for the characteristics of high-density culture. So these cells will reach greater than 20 million cells per mil in shake flask cultures. They have a very rapid doubling time, so about 17 to 18 hours. So any of you that have worked with them, realize that if you come in the morning and your ExpiCHO cells are not at the density that you need them to be, you need only wait a few hours, and they will be where you need them for your subculturing or expression purposes. The cell diameter, it ranges from about 14 microns in routine culture to about 20 microns during expression, so the cells do become very large. And again, we do have high transfection efficiency in the 75 to 80% range with the expifectamine CHO transfection reagent. We've got stable growth and expression profiles for at least 20 passages for the ExpiCHO cells, keeping in mind that because the doubling time is so much shorter for the ExpiCHO cells, that 20 passages represents a significantly larger number of population doublings compared to the Expi293 cells. And what we've shown is that we tend to get CHO-like glycosylation profiles that match the protein being made in the stable cell line. And once again, very high quality biologically active protein. In the bottom, you see the ExpiCHO cells in the middle pane at 6 million, the right pane at 20 million, and even at 20 million, there's essentially no cell clumping. So that is one thing that we always want to keep an eye on. The cells should not be clumping in any of your cultures, and if they are, there is further optimization that we can work with you to alleviate any of those issues. So if we look at the actual growth kinetics, on the left you can see again Expi293 cell growth. So these are just cells seeded at 500,000 cells per mil, allowed to grow in shake flask cultures. And you can see in the red line is the viable cell density. And typically for Expi293, we would transfect at 3 million cells per mil. And we typically split the cells between about 3 to 5, when they reach 3 to 5 million cells per mil. These cells in this case peaked at about 15 million, as you can see, and the blue bar across the top is the viability, close to 100% across that time. Once the cells reach their peak density, they will crash, as you see, going from day 6 to 7, and the viability will drop accordingly. On the right, you'll see the ExpiCHO cells, in this case growing up to over 20 million cells per mil in the shake flask cultures before they crash and the viability and the density drops. ExpiCHO, we transfect at 6 million cells per mil, and typically we recommend passaging the cells when they reach 4 to 6 million cells per mil. Although with the ExpiCHO cells, and in fact with both cell lines, it's preferable to overgrow those targets rather than undergrow them. So you do not want, for either of the high-density cell lines and high-density cell lines in general, they tend to not like being split before they get into that early log phase growth. So if you do have an instance where the cells overgrow, even significantly, in the case of ExpiCHO, 10 million or even higher, we recommend simply splitting them back more gently to about 500,000 cells per mil, and the cells should recover perfectly fine. We have seen instances where when cells were over-subcultured at low densities for many days in a row, for example, when a transfection couldn't take place and the cells were cut back once or twice, at times those cells will not recover for you. So again, it's very important to get them into that log phase growth before you do your subculturing. And all of those work towards having the best possible passage stability. If we look at the complexation, the transfection reagents of the two kits are very different. For both of them, we've found great utility with pcDNA 3.4 vector. For Expi293, typically 1 microgram per mil of plasmid DNA is most optimal, whereas ExpiCHO somewhere in the 0.6 to 0.8 micrograms per mil has been shown to be optimal. And both of them use the same 3.2 microliters of transfection reagent per mil of culture to transfect. For Expi293, OptiMem is the recommended complexation media, and for ExpiCHO, it's OptiPro serum-free media. For Expi293, typical complexation timing is at least 5 minutes and up to 20 minutes, where you see good stability in the figure below. Whereas for ExpiCHO, having a more powerful transfection reagent to transfect the CHO cells, we typically see complexation times of essentially 0 to 5 minutes. So for ExpiCHO, the nice thing is that we do the complexations with cold reagents, so you can literally pull your reagents out of the refrigerator, dilute your plasmid DNA first, we recommend, then dilute your expifectamine CHO transfection reagent, add them together and mix by gentle inversion or 1 or 2 gentle pipettings, and you're actually ready to transfect your flasks right away. So probably a total time of maybe 5 minutes start to finish for the complexation and transfection in ExpiCHO. And I will get into later strategies for automation, given the differences in the two complexation reactions. And again, what I mentioned earlier is that very important that the feed-in enhancer, so the utilization of the entire Expi293 and ExpiCHO system is critical for getting that biggest cost versus titer benefit of the systems. So on the left, you can see Expi293. When we add the enhancers, we can more than double the protein titers, and even more dramatically on the right in ExpiCHO. So again, when you look at the control scenario here, if you simply transfect ExpiCHO cells in ExpiCHO media, they will overgrow, and they will make very little protein as they use all of their energy for growth, and they will eventually crash within a couple of days. So again, here, when we add our feed and enhancer, in this case, we get many-fold improvements in the titer compared to transfecting only the cells. When we add the feed and enhancer is when we can typically get to the multi-gram per liter titers for many antibodies. So again, very important to utilize the entire systems. The benefits will outweigh the costs. When you utilize the entire kit, you will get a significant improvement in your cost per milligram of protein expressed. When we look at the heavy chain to light chain ratio for antibodies, we typically see in the 293 cells that a 1:2 ratio of heavy chain to light chain is optimal for Expi293, versus in the CHO cells, we see that a 1:1 ratio of heavy to light chain seems to be optimal for the ExpiCHO system. So one slight difference there. Our positive control rabbit IgG antibody that we provide as part of the starter kits or make available separately is a 1:2 ratio per Expi293, but you can see in ExpiCHO on the far right, while the 1:2 ratio is not as good as 1:1, it still does give significant titer, and thus the positive control is suitable for use with both systems. When we look at the plasmid DNA titration in Expi293 versus ExpiCHO, there's significant differences here in that in the 293 cells, we recommend typically about 1 microgram per mil, but even a bit more, even up to 1.2 micrograms per mil of plasmid DNA per mil to transfect will give a bit of a boost in titers. Whereas if we contrast this to ExpiCHO, about 0.6 and 0.8 micrograms per mil is the peak DNA concentration in the ExpiCHO system, which is quite interesting in that we are transfecting twice as many cells in ExpiCHO per mil as we are in Expi293, yet because of the exofectamine CHO transfection reagent, we're able to use significantly less DNA. And here you can see on the right, again, even at 0.4 micrograms per mil of DNA, we're not losing significant titer in the ExpiCHO system. So again, a difference between the two different cell lines and transfection protocols. When we look at the workflows, Expi293, for both of these, we recommend the day-1 subculture to give the cells fresh media to provide the highest titers from the systems. For those of you that are doing very small volumes in the Expi293 system, you can actually spin the cells down on the day of transfection and dilute them in 100% fresh media to the 3 million cells per mil for transfection, and that will give you probably in the area of 10 to 20% higher titers. That becomes less possible to do as you scale up, but many labs that are running at small scale will employ a full media exchange at the time of transfection to provide as much fresh media and as high titers as possible in the small scales. So we have a day 0 transfection at 3 million cells per mil, again looking at the Expi293 workflow. 1 day later we add enhancer 1 and 2. For both of the systems, the enhancer 1 and 2 can be pre-mixed ahead of addition so that you're only adding one time rather than adding enhancer 1 and 2. And in fact, we have data to indicate that you can mix enhancer 1 and 2 together ahead of time, keep them at 2 to 8 for even up to a month, such that you can only do a single addition of the enhancer and feed instead of 2. And then typically harvesting on day 5 to 7 for Expi293. In contrast, the ExpiCHO workflow, we subculture at day -1 to about 3.5 to 4 million cells per mil. Those cells will typically grow overnight to 10 million cells per mil or higher. We'll then dilute them on day 0 down to 6 million. We add on day 1 the enhancer and the first feed. And then at this time, you have a decision point whether to go remain at 37C for the standard protocol or to shift to 32C for the max and high titer protocols for maximum protein expression. On day 5, if using the max titer protocol, we would add the second feed and then typically harvest on day 7 for the standard protocol, day 7 to 9, and around day 10, 12, potentially as far as 14 for the max titer protocols if you're looking to generate maximum protein titers. So the one thing that's interesting between the two systems is how the cells make their protein. So Expi293 cells tend to be, if you look at the red curve, they express quickly, but they also fizzle out quickly. So Expi293, usually around day 5, 6 is when you're going to reach peak titers. The cells do start expressing a little bit faster than the CHO cells. But the CHO cells you'll see in the 3 blue lines, so the dark blue on top being the max titer, followed by the high titer, and light blue standard titer. These protocols, if we look at the max titer protocol, on that day 5, 6 of harvest, ExpiCHO will actually express at very consistent levels as Expi293. So we often get the question around whether or not the Expi293 expression kinetics are faster or better than CHO, and in fact in many instances it seems that they're quite comparable at the time of harvest for Expi293. However, ExpiCHO allows you, as you can see from day 6 to 12 or 14, you can get about another 2/3 of protein compared to where you are at day 5, 6. So that really is that additional benefit of the CHO cells and that characteristic of them that they can continue to make protein for such a long period of time. However, as you see in the upper right, one does not always want to go out to 12 to 14 days depending on the type of protein that you're expressing. Again, Expi293 typically 5 to 6 days for stable proteins, monoclonal antibodies, secreted proteins, whereas ExpiCHO probably 7 to 12 days is a sweet spot for those proteins. Whereas in both systems, membrane proteins, typically we will harvest in the 48 to 72 hour range. You find the best expression at that time. And then intracellular proteins would be somewhere intermediate of the secreted stable proteins and the membrane proteins. So I think it's important to understand what your need is with the protein that you're expressing, and then harvest at that appropriate time. If it's a less stable protein, you're going to want to harvest earlier on. If it's a very stable antibody where you're looking primarily for the ability to bind to its target, you can take ExpiCHO out 12 to 14 days and make as much protein as possible. So one of the other differences between the two systems is the clarification of the supernatant and purification. The CHO supernatants can be a little bit more difficult to work with, simply because the componentry for CHO cells is somewhat different than Expi293 cells. So generally in ExpiCHO on the right, we would recommend to spin the supernatants at 4,500 to even up to 7,000 times G for 30 minutes, 2 to 8. This will significantly clarify the supernatant and make it much easier to filter. Both of these we would then put through a 0.2 micron PES filter. And then we typically use for protein A purification tris buffers, and there's an application note on this for ExpiCHO. And then for ExpiCHO, often these proteins and antibodies may be going into animal studies, etc. So an ion exchange polishing step, or SEC, is often employed in the ExpiCHO purification schematic. So again, here just representing that from the culture supernatant, the faster you can spin, the better for ExpiCHO. We go through the bottle top filters or depth filtration depending on the volume that you have. Typically going through for antibodies the protein A traps with either a tris or a HEPIS-based binding buffer. We have found that some of the components in the ExpiCHO system can stick to the protein A columns when using phosphate buffers, so our application note provides alternatives for cleaner protein A elutions. And then again, CEX or AEX polishing steps. For his tag proteins, many of you that have worked with them know that some of these higher density chemically defined medias can impact his tag purification. So there are a number of strategies that can be employed to improve purification in Expi293 and ExpiCHO. One is to increase the resin to media ratio. Secondly, to dialyze the supernatant into a compatible buffer. Thirdly, employing a strip-resistant nickel resin such as the Excel resin is a good alternative. And then lastly, the potential to consider an alternate protein tag. So later today, we'll be joined by a scientist that will give a presentation, Dennis, on the streptactin system, which is an excellent alternative to nickel NTA and his-tagged protein purification. So among all of this, the keys, again, to these systems is simply matching the volume of culture to transfect with your flask size, your shake speed, and your orbit. So when we think about the four things that need to be optimized for any expression system, we want to make sure that our volumes of media as well as flask size are matched to the speeds and the orbits of our shakers. So that is critical. All of these systems are high density, so they push the limits in terms of cell capacity, and it is important that, as a start, these conditions are adhered to, and then as you have more experience working, you certainly can tweak these in different ways in your labs. But for the purposes of getting started, we do recommend to follow these conditions that are in the protocols quite closely for your initial expression runs, because that is absolutely important for optimizing the systems. And then moving out of shake flasks, so the mini bioreactor or the spin tubes, these are 50 ml tubes that have a gas permeable membrane on them, very popular with many labs. You can see here data for ExpiCHO using the standard protocol or High-Titer with final volumes of 20 to 25 mLs in the tubes, very comparable to the 125 shake flask controls in red, and the conditions on the far right for the 19, 25, and 50 millimeter shakers. And these conditions can be utilized as well with Expi293. The only caveat with ExpiCHO, because of the higher cell density, as you get to these 20 or 25 mil volumes, you will need to tilt the angle of the tubes by about 15 degrees to keep the cells in suspension at these shake speeds. And then next, the 24 deep well plates have become an absolute workhorse of the protein expression community. With the Expi systems, one can now generate a fair amount of protein in 3 to 4 mLs. That was not possible previously. So these have really become workhorses. The 24 deep well plates, we typically recommend the ones that have the square or the pyramid bottoms. Round bottoms also work well, but we and others have found that the pyramid bottoms tend to be a bit more robust and give a little bit better results and viability. The cells actually seem to really like these plates. The viability tends to be as high or even higher than the shake flasks. And again, shown here for ExpiCHO, the control 125 ml flask versus the 24 deep well plates. The standard or High-Titer protocol is getting very comparable titers. And in the table to the right, you'll see again the shake speeds per the different shaking diameters, transfection volume, and the final volume in the wells. And then lastly, the 96 deep well plate option. So this provides about 1 ml of final volume, shake speed of about 900 to 950 RPM. And what's important here is that the shaking diameter of 3 millimeters, it needs to be an orbital diameter. So some ELISA plate shakers will shake in a horizontal axis. It is important that the shakers are 3 millimeter orbital, and again, you can see highly comparable titers compared to the 125 ml control flask for the 96 deep well plates. And for all of these, the last 3 scales that I mentioned, we do have an application note online with all of these conditions, so you can refer to that for your scale down protein expression needs. In terms of automation, I'll just take you through this very quickly. For Expi293, the DNA transfection reagent complexes are more stable, so they can typically sit around for 20 to 30 minutes on a deck. For ExpiCHO, because of the quick complexation, we had to alter the protocol a bit. So we used our Amber 15 just as a test case for automation. And what we found, and this is actually quite handy even on a day-to-day basis, is that What we do here is that we doubly dilute the plasmid DNA, so in the red panel up top, we doubly dilute the plasmid DNA with the full volume of OptiPro that you would normally use to dilute the plasmid DNA and your transfection reagent. So we doubly dilute the plasmid DNA, and that's very stable, so that can sit for long periods of time. And then we would have the robot actually add the neat ExpiCHO transfection reagent directly to the doubly diluted plasmid DNA. We would mix that twice on the robot with the tips, and then immediately add the complexation, transfect the cells in the vessels. So in this way, we're not pre-complexing and allowing the complexes to sit on the deck. We doubly dilute the plasmid DNAs, and then we add the neat ExpiCHO right at the time of complexation, mix 2 times, and transfect our cells. And in fact, this is the same protocol that we suggest in all of the scale-down protocols so that you're not working with such small volumes. You can dilute out all your plasmid DNAs first, add the ExpiCHO neat to them, and you will have equivalent titers. So many labs actually do this, what we call a one-tube complexation method for all of their scales, just because it allows them to dilute their plasmid DNA. That can sit for as long as you want, and then just add your neat transfection reagent at the time of complexation. So breaking into some of the expression data, what I did want to note here, this was data generated internally using Expi293 and ExpiCHO max titer protocol for a panel of rabbit monoclonal antibodies. And what we see in the figure, so Expi293 is in the red bars, ExpiCHO is in the blue bars, and that we generated about 1.4 to 3.9 fold improvement in ExpiCHO versus Expi293 for this panel, with a mean of about 2.4 fold improvement over Expi293. One antibody, actually MAB19, did not express at all in Expi293, but we still got close to 200 mg per liter in ExpiCHO. And then one antibody, actually number 20, didn't express in either system. And that's unclear as to why at this time, but certainly you can see all of these rabbit monoclonals share the same backbone with the exception of their CDRs, and you can see the significant differences solely in the impact of the different CDRs in the expression levels of these proteins. And then to help with the detection of your proteins of interest early on, in the June to July timeframe, we should be launching these ProDetect lateral flow strips. And in the bottom left, you can see all the different tags that they're directed against. And these strips, typically, if we look in the assay setup, it's about 10 to 15 minutes in your culture supernatant. In the case of the middle pane, you see a competitive assay for MICTAG, and to the right of that, a GST assay. And you can see the test line that shows up when you have a positive response. And in the far right, these are strips that we actually processed in Expi293 supernatant for rabbit IgG. And as many of you know, our positive control for the systems is a rabbit IgG. So these strips will provide a very easy way for you to see visually the expression of the rabbit IgG in your systems. So they have a linear range of detection of about 10 micrograms per liter up to 1 mg per liter in crude supernatant. So this is all done in crude supernatant. And at the higher concentrations, for example, the 100 mg and 10, you see that they're, like most immunoassays, you do get a bit of a hook effect. Where we found great utility for these is that when we're expressing proteins for the first time around and we don't know how well they're going to express, or that class has a history of low expression for given constructs, we can quickly screen the supernatant at small scale to ensure that we're getting good protein expression. And then, maybe that's 1 to 2 days into a small scale expression run, and then we can then very quickly scale that up as opposed to waiting 5 to 7 days, generating titers, and then scaling up. We can probably cut almost a week off if we know that we're making protein at the small scale. We can take those constructs and then scale them up. And then looking at, from that panel of top biologics that I mentioned earlier on, these are 4 different biologics. So they Herceptin, Avastin, Enbrel, and FC Fusion, and you can see as real-world examples, these molecules expressing, close to or above the gram per liter range in ExpiCHO for some of these legacy biologics. In terms of his-tag proteins, this was done in ExpiCHO. You see on the left panel 2 different FABs. So you see non-reduced, very nice quality of the FABs, and reduced at about 25 kD using an anti-his-tag Western blot. In the middle, you'll see 2 C-terminally his-tagged proteins, again showing up nicely from ExpiCHO, and then N-terminal his-tags on the far right. This slide is a case study of one of the labs that utilizes the ExpiCHO system, and it speaks a little bit towards optimization and those parameters that I mentioned earlier. So in their very first assay, they still got reasonably good titers of their protein. We had a few discussions with them. They did a bit of vector optimization on their own, and then we simply made sure that the transfection processes and the cell culture best practices were being adhered to. And from that first assay on the left to the optimized protocol, we achieved almost an 8-fold improvement. And on the right, you can really see the power of the Expi systems versus the older HEC293 or CHO-S systems, where for these 2 proteins, 1 and 2 non-antibody proteins, 750 to 800 mg per liter versus a maximum of 11 and 60. And then for the human IgGs, 950 and 360. And again, for human IgG number 2, no expression at all in the legacy CHO-S system. So similarly, this was for FAB fragments. On the left, again, is this bump that they were able to achieve by optimizing. And then you can see on the right-hand side, very thick bands, so they were eventually achieving greater than gram per liter levels of purified Fabs from the ExpiCHO system. In terms of GPCR, so this is some data around a GPCR expressed in pcDNA 3.0, harvested at 72 hours, and in both cases of Expi293 and ExpiCHO, 32C was shown to be better than 37C. We didn't see any impact on cell viability, even if we let these expressions go for longer periods of time. And in fact, the expression levels were quite comparable between ExpiCHO and Expi293. If you look at the bottom left, the E. coli reference protein at around 37 kD that had been used, and then lanes 2, 3, 4, and 5 are Expi293 with different amounts of plasmid DNA, and similarly 6 through 9 for ExpiCHO. And you can see in terms of activity, both Expi293 and ExpiCHO are comparable or better on a per mass basis to the E. coli control material that had been used previously. Similarly, we did some work on an ion transporter, and this was expressed in Expi293 or ExpiCHO, quite difficult to express. Here we looked at different promoters, as well as different concentrations of DNA, whether we had a 3 or 5 prime tag or 37 or 32C. And here, interestingly, if we look at the bands, the second and fourth band under Expi293 on the left-hand side, you see very strong expression of that protein, actually at 32C, again, being better than 37C, even for the 293 cells. Whereas we actually got very poor expression in ExpiCHO. And one question that I had here is that this transporter is of specific to neurons, and with Expi293 having a neuronal origin, it is intriguing to consider whether or not that may be why, for this particular membrane protein, the Expi293 cells do a much better job than ExpiCHO. So membrane proteins are always tricky. They tend to be different from one protein to another and require optimization, but these are two interesting examples of some similarities and differences between the Expi systems. Then in terms of vaccine development, just a real quick story, a group out in UCSD led by Jiang Zhu is generating these trimeric and very large HIV nanoparticles as vaccines. And just a snapshot of the data from their paper, typically these were all generated in 293 cells, and Dr. Zhu took these and put them in ExpiCHO. And what you see on the third panel from the right in red is the ExpiCHO expressed particle. And you see a single peak versus the 2 peaks of Expi293. And on their first go-round, they were able to generate 25-fold higher particle mass in ExpiCHO versus Expi293. And then we had a visit with their lab. We made two small suggestions in terms of their shake speed and temperature, and they were actually able to get up to 60-fold higher protein expression in ExpiCHO versus Expi293. And they're also showing that the antigenicity of the ExpiCHO-derived material is in fact superior to the 293 material. And these particles, they are huge, and about almost 50% of the mass is glycans. So these represent very difficult, large protein complexes that historically had been made in 293, but showed significant benefit in the ExpiCHO system. So I will just talk for a few moments about the training materials and resources that are available both online and virtually. As I mentioned, for ExpiCHO, there is an essential protocol guide, as you see on the left, that has much of the frequently asked questions and tips for getting started in ExpiCHO captured in that document. We also have the guide to optimizing protein A purification of monoclonal antibodies in ExpiCHO, as well as, I mentioned, the protocols for the 24 and 96 deep well plates and the mini bioreactor tubes for ExpiCHO online. The other resources as well is that if you go onto the Expi293 or ExpiCHO website, or even YouTube, and you look up Expi293 or ExpiCHO on YouTube, there are also instructional videos online made by Xiaoyan Liu, who's our lead scientist on the Expi systems, showing exactly how our labs are set up, exactly what our protocols and how the transfections, cell handlings, etc., are performed in our lab, and they truly are about 6 to 9 minutes, but they are very educational in terms of seeing exactly how we are doing things in our lab where the systems were developed. And then one of the newer things that we released very early this year is our Expi293 and ExpiCHO online protein expression protocol calculators. So you can see the URL in the upper right here. And what these allow you to do online, you can go in, and if you look at the bottom left panel, you can select whether you're running ExpiCHO or Expi293, and each of these tabs will walk you through the preparation of the cells in terms of what flasks, volumes, orbits, shake speeds, etc., transfection of the cells, the addition of the enhancers and feed, and then you'll have the ability to print this out in a notebook copy that you see on the bottom right, which will have all of your conditions for preparing, transfecting, adding the enhancers and feeds for reference during the actual performance of your experiments. So a very handy tool to utilize to get all of your volumes in one place for your experiments. And then lastly, I wanted to also take a moment to point out some of the services and support that we offer in terms of the LabCoat Live virtual training courses. So as you see in the upper right, they have the ability to register for these courses. And these courses essentially are virtual, so there are trainings that take place in a virtual environment, and the steps would be followed in your own lab along with the guidance of the instructors performing the side-by-side experiments in our internal laboratories. So this is a great way, if location-wise it's difficult for you to visit our labs or for us to visit your labs. These are excellent interactive ways to get up and running with the systems with a virtual training. But again, I do highly recommend to utilize the resources online, especially the videos, because I think as they say, a picture is worth 1,000 words, and videos are worth much more when you can actually see all of the steps and exactly how they are performed in our laboratory, our equipment, etc., which sometimes can shine significant light on similarities and differences in the way protocols are being performed. Lastly, I will just take a moment to provide a special thanks to all of our Thermo partners in protein expression R&D, product management, marketing, and our field application scientist teams. Many of you on the line, I'm sure, over the years have interacted either by email, telecom, a video conference, or many also in person with myself or my team. On the left, you see our R&D team. Can't thank them enough. Many of you know Chao Yen, have met with her extensively at the conferences. A real wealth of information for all of our collaborative labs. And many of you also know Henry Chu, who has been in the protein expression field for many years now. And thanks again. We recently have brought on 2 field application specialists, one in the EU region, Nanyang, and Matt McKenna supporting the North America based in the Boston region. But these field application scientists, when my team is not available, they tend to be available for in-person consultations or, again, telecoms and answering of email questions. So I would like to thank everybody there. And with that, have about 10 minutes left in the hour, and I will wrap up and then allow for any questions that you may have in the audience. And thank you once again for your attendance today.
Thank you, Dr. Zmuda, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window. Type your question into the box that appears on your screen and click the Send button. We'll answer as many of your questions as we have time for. So let's get started. Our first question is, Expi293 is typically harvested on day 6 to 7. Do you need to wait until day 12 to 14 to harvest ExpiCHO?
Great, thank you, Alexis. Excellent question. I think I touched on this a little bit earlier. I would say that, again, for ExpiCHO, going out to day 12 or 14 is one of the benefits of the system. If you have the time to do so, you can generate very high-titer antibodies or stable secreted proteins. But at the same time, it's not always necessary to do so or even desirable. So again, for both of the systems, less stable proteins, sometimes coagulation factors, for example, or membrane proteins, intracellular proteins, you would tend to harvest them earlier on. But even the stable secreted proteins in ExpiCHO, if you were to harvest at that same time as Expi293, day 6, 7, 8, the titers in ExpiCHO will typically be comparable and in some cases even higher. So it's not always necessary. I think we should not think about ExpiCHO that you're forced into a 14-day protocol. You're still going to make very high levels of protein even if you harvest earlier. And then again, to keep in mind the different classes of protein that you're expressing, because many of them, in the case of both systems, you will not want to push them out to those latest time points due to potential quality concerns of the proteins themselves.
What is the most important indicator of optimal performance of the ExpiCHO system?
Yeah, a great question. And again, as we look back to some of the slides that I showed around the key for setting up the experiments, we can get back to them here. So this table, again, when you're getting started, important to follow the guidance provided in the protocols. And for ExpiCHO, the key thing that you want to look at is that on about day 4 or 5 post-transfection, if your viability starts to drop off, that tends to be the primary indicator that something is just not quite right in terms of the culture conditions. So the ExpiCHO cells, even if the conditions are not right, will hang in there for 4, 5, 6 days, but then the viability will start to drop. So what you really want to see in ExpiCHO to know that you're optimized with your conditions is that you've got that high viability throughout the expression run, so that even on day 12, 14, you may be in the high 70s or 80% viability. If you start to see, again, that drop, around 4 or 5 days, almost exclusively that indicates that, some parameter simply needs to be tweaked a little bit, and once done, that high level of viability will be maintained, and the cells will be healthy and express high levels of protein.
Dr. Zmuda, our next question. What is the best strain for A/B screening experiments?
So, again, both 293 and CHO have been used for early candidate screening, and they still are. I would say that the momentum is moving more in the direction of CHO. So many biopharmas are interested in starting with CHO and staying there throughout the entire process, which is also part of the reason that we've offered the GMP ExpiCHO cell banks as well as the stable production media for those labs that do their initial screening in ExpiCHO and want to follow their development of that molecule in the ExpiCHO cells all the way from start to finish in terms of clinical material manufacture. So I think both of them are good. I think if you're screening human antibodies, they're both good choices. If you're screening murine antibodies or rat, as I mentioned, you really want to move in the direction of ExpiCHO. And then again, ExpiCHO, depending upon the post-translational modifications that may be important to you and or the ability to generate higher titers in smaller volumes at that screening stage, may be one reason that you would look to go to ExpiCHO versus 293. I think in general, ExpiCHO does a better overall job of expressing antibody molecules across the board, whereas the 293 cells can on occasion run into some antibodies that they don't like to express or they express at very low levels.
What guidance can you provide for which system to use for expressing different classes of proteins? When to use Expi293 versus ExpiCHO?
So another great question. Empirically, it's impossible to say. We don't have a database of all proteins out there that have been tested in all the systems. But I would say in general, what we're seeing, the industry trends, is that anything that is an antibody or an antibody-like molecule, a fragment, a fusion, very successful at high titers in ExpiCHO. Again, the murine and rat antibodies. Expi293 still tends to be very popular for making reagents, ligands where you want to have human-like post-translational modifications, but they also do a very good job in antibodies and fusions as well. Lastly, I think in general, Expi293 seems to still be the go-to for structural biology, membrane protein expression. I think laboratories are very comfortable with those cells, as well as intracellular proteins, although they tend to be split fairly consistently between the two systems. So it really is, hopefully a database of knowledge can be built in your laboratories, but oftentimes antibodies and clinical therapeutic candidates in CHO and reagents, structural biology, et cetera, in 293.
And our next question, what would happen if we use 293 T cell instead of Expi293 cell?
So, great question. The 293 typically is an adherent line. There are some that have been suspension adapted. Most likely, if you could get the 293T suspension adapted into the Expi293 media and growing, I think that it's likely that you would be able to get reasonable expression, probably still not anywhere close to with the entire Expi293 system, but that is a possibility. The question that I would ask up front is there an absolute necessity to move to the 293T line if you can demonstrate comparability between the T and the Expi293 and you are seeing better results and higher titers in the Expi293? That may be a better way to go, also because of the fact that we have the GMP Expi293 cells banked, that if it became an item where you were looking to move this into clinical manufacture, those cell banks would be available for licensing opportunities.
Dr. Zmuda, what would happen if we incubate the cell without shaking? We do not have orbital shakers adjusted by our incubator.
So here, unfortunately, the answer would be that you would not want to try this. The cells, being suspension cells and high-density cells, they need to be maintained with agitation in the shake flasks such that we get proper gas and nutrient exchange for these high-density cultures. Now, for gene editing, things along those lines, you can actually add some serum, and these cells will stick down readily to tissue culture plastic, and that can have some utility for editing. But typically, you would need to have the shakers to maintain the cells and the performance of the cells.
And it looks like we are out of time for questions, but I would like to once again thank Dr. Zmuda for his presentation. I would also like to thank Thermo Fisher Scientific for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through May of 2019. Please share that with your colleagues who may have missed today's live event. Also, stay tuned for our next presentation, a chemically defined baculovirus-based expression system for enhanced protein production in SF9 cells, presented by Maya Yocheva. You won't want to miss it. That's all for now. Thank you for joining us, and we hope to see you again soon. Goodbye.